Jonathan is our last but not least, as I said, and is another match made in heaven with a mentor, since the application is like I love physics and I love chemistry. So what can be better than work in the light microscopy core? And actually, I have to say, it was one of the first times that I had to actively go to find an intern in the lab and tell them that it was too late and it was time to get out and go home. Jonathan.
Hi, my name is Jonathan Huss, and I'm a rising senior at Donna Klein Jewish Academy. This summer, I had the pleasure of learning under the guidance of Katie Shirley and Dr. Urban from the Light Microscopy Core, and my project was on the optimization of the Lightsheet imaging pipeline via comparisons between tissue clearing methods and data analysis softwares. So, currently, at the Max Planck Florida Institute for Neuroscience, many researchers use tools such as the two-photon, confocal, or STED microscopes in order to view very tiny structures within the brain. Now, while these scopes are effective, they often take up large amounts of time as they are limited to illuminating only a single point within the tissue, and they can only penetrate deep enough in order to image very thin slices of tissue. Now, imagine that you're a researcher and you want to image a whole brain on the confocal microscope. In doing so, you would first have to spend a couple of hours simply slicing around 50 or so slices. Then, you have to spend around a whole week solely imaging, and then another couple of weeks processing and analyzing the images. That rounds it off to about a month before you can even begin collecting data from your images and render the whole brain. Now, to me, that sounds pretty tedious and like a misuse of precious time that could be used toward advancing the field of neuroscience. Now, as a growing number of those who desire to view the whole brain have yearned for an improved system, a novel machine was thus developed. Here at MPFI, we recently received the Zeiss Lightsheet 7. It has been increasingly used and thus has made its way to revolutionize the field of microscopy. By illuminating a whole plane within the tissue instead of a single point like from the previous scopes, the Lightsheet is able to collect whole brain 3D renderings in as quick as 5 minutes. Here on the right is a short example of one such rendering, where there are clear retrograde trace cells within the insular cortex of the 3D rendered brain. So you may be wondering, though, how does the light pass through the whole tissue so that we can acquire these high-quality images? This is where tissue clearing comes in. Tissue clearing really does what it sounds like. It actively renders the given tissue transparent in order to allow light to penetrate more deeply into the sample and allow for high-quality imaging. As seen in the above figure, the tissue begins opaque, and then after the tissue clearing process has finished, it ends clear. In addition, there are a number of different clearing protocols that achieve this clearing effect, such as cubic, UDisco, FDisco, and many others. However, the key takeaways here are that they each destroy a different number of targeted biomolecules that a certain researcher may be interested in. They each either shrink or expand the brain to a certain degree, and they each leave behind a varied level of clarity within the tissue, thus making it difficult to discern which protocol is most viable to view whole brains. So now that we have a good basis on the Lightsheet and the role of clearing, we can now move towards the bigger picture. In viewing the Lightsheet pipeline as a whole, from clearing to imaging, data analysis, and then getting your final result, two problems arise. With the overwhelming number of clearing methods and data analysis softwares, it's difficult for researchers to point to an optimized and reliable pipeline that yields high image quality and data analysis results. At this time, many researchers may be unaware of the many alternative clearing methods and data analysis softwares. However, as there are a number of them both, the Lightsheet pipeline can be further optimized in order to optimally visualize and analyze whatever one lab may be interested in. Our role as the Light Microscopy Core is to investigate these knowledge gaps and provide the researchers here in the Institute with the best possible flow for achieving quality data. Therefore, I sought out to efficiently optimize the Lightsheet imaging pipeline in order to make known the Lightsheet's versatility, ability to create high-quality images, and time-saving aspects, while also creating a properly established formula that those of you already using the Lightsheet can follow. Now, in order to accurately refine the Lightsheet pipeline, I began with modifying its first step, to tissue clearing, a head-to-head comparative approach between 4 popular clearing methods used here in the Institute: EasyView, FDisco, iDisco Plus, and UniClear, was utilized in order to uncover which protocol yielded the best results. While many aspects of each clearing protocol could be tested, I chose to focus on the most important point, the clearing ability. This was achieved through performing the various clearing methods and then qualitatively and quantitatively comparing their clearing capabilities against one another. In analyzing the above images from a simply qualitative standpoint, it can be understood that EasyView cleared to the greatest extent as the structures within the midbrain of the tissue were most clear and visible to see. In addition, UniClear was seen to be the runner-up to EasyView, as with a little less light penetration, but the structures within the midsection of the tissue were still visible. iDisco Plus was noted to be mediocre in its clearing ability, and FDisco was noted to be the least effective, with the least light penetration and the tissue being left with the midsection of the tissue being left opaque after clearing. Furthermore, quantitative observations were drawn by measuring and graphing the light intensity profiles in the midsections of each cleared brain, and then running the values through a one-way ANOVA analysis. Whichever clearing method yielded a brain with the greatest average light intensity therefore worked best, as it was able to allow the light to penetrate more deeply into the sample and allow for higher quality imaging. As seen in the graph above, the x-axis represents the various clearing protocols, and the y-axis represents the intensity. Furthermore, the intensity values were standardized to fit between a 0 and 1 range. Now, the middle bar represents the average light intensity, the top bar represents the maximum light intensity, and the bottom bar represents the minimum light intensity. Now, together, they represent the total light intensity profile of the cleared brain's midsection. Now, as we can see from the figure above, EasyView presented with the greatest average intensity and also presented with a similar range of intensities throughout the midsection. Furthermore, UniClear came close with a similar average intensity throughout the tissue, with a similar average intensity, however, a larger distribution of intensities throughout the whole tissue. iDisco Plus was noted to be mediocre, as seen from the images before, with a little lower average light intensity, and FDisco was seen to be the least effective, as it had the lowest average light intensity. So, now that we know EasyView performed the best in regards to tissue clearing, and is thus the most optimal for imaging on the light sheet, we can now move on to the second set of comparisons between the analysis softwares Arrivis Vision 4D and Amaris 10, in order to better optimize the back end of the light sheet pipeline. The two softwares were tested for their functionality, process timing, ease of use, capabilities, and ability to output proper data. To begin, the Amaris 10 software offers a user-friendly, step-by-step guided pipeline in order to achieve neuron tracing. However, there is little to no customization options, and thus the user is left constricted in only tuning the software's given parameters. However, even with this constriction, results such as those expected here on the left were actually realized by our dataset on the right. Now, as we can see, the neurons were traced nicely with little registered artifact. In order to achieve this output, the user must run the dataset through multiple sets of training and hand validations. In total, the output takes around a couple of hours for fine-tuning and training, and is pretty straightforward in terms of ease of use for the user. Now, while Amaris 10 is able to accurately trace these whole neurons, its functionality for our research purposes ends here, as it is unable to handle large data volumes such as those we receive from the light sheet microscope. However, in using Arrivis, it instead offers a customizable analysis pipeline, as seen above, that can then be fitted to many different types of data sets. For instance, probe tracking, cell counting, and neuron tracing were found to be very effective in the Arrivis software. The software also offers very advanced deep learning and machine learning training tools so that the user can outline a series of predefined objects, such as cells, to then feed into the algorithm, and then for it to then spit out the rest of the location of the other cells. One can thus see the segmented results I received after only spending minutes creating the pipeline and a couple of hours in training. Here on the left are clearly segmented cells, on the right are clearly segmented neurons, and on the bottom are clearly segmented probe tracks. With only minutes of fine-tuning a pipeline, Arrivis can offer accurate segmentations that can then be applied to other whole data sets. In short, It offers a versatile analysis environment that can trace virtually anything the user would like. Furthermore, the software houses many other desired features, such as a high-quality tile stitcher, a robust data transformation option, and the ability to warp and register whole or partial brains to the Allen Atlas. So to tie these findings together, I would like to reiterate the main question of which clearing method and data analysis software could be utilized in order to gather high-quality Lightsheet data to thus further explain the gravity of my findings. In performing these head-to-head comparisons, I found that not only can the whole brain imaging and data analysis be realized in a fraction of the time using Arevis and the Lightsheet, but the ambiguity of the clearing effect of the various protocols was thus lifted in discovering the capabilities of the various clearing methods. Therefore, I propose 3 Lightsheet pipelines for accurate whole brain cell counting, probe tracking, and neuron tracing. For both cell counting and probe tracking, I propose a Lightsheet imaging pipeline that entails clearing with EasyView, then imaging on the Lightsheet, and then performing data analysis with Arevis. As it was concluded that EasyView was most effective in clearing and Arevis was very successful in completing both segmentation tasks, this is thus a well-optimized pipeline that can clearly yield high-quality data results. With neuron tracing, the outlined pipeline would be as follows. It would begin with EasyView clearing, then imaging on the Lightsheet, and then using either Arevis or Amaris-10 for the segmentation. So Arevis and Amaris-10, as Arevis and Amaris-10 presented similar neuron tracing capabilities, further testing would need to go into determining which one could produce a better output in a shorter amount of time. So now that we have these pipelines, what's next? The project does not end here, and we could further optimize the clearing protocols themselves to allow researchers to not only clear their tissue, but also customize it with their dyes of choice for immunolabeling. We can also begin by formulating a clearing protocol index that catalogs each protocol's effectiveness, their costs, their timing, and to thus point researchers towards the most optimal method for their research goals. And lastly, we could optimize the Lightsheet imaging pipeline by testing different sampling rates for the fastest imaging time while maintaining quality images that can still be segmented by the data analysis softwares. So with this, I would like to thank my mentors, Dr. Urban and Katie Shirley, for being the greatest mentors I could ever ask for and for bestowing upon me the knowledge of microscopy and neuroscience that I'll take with me along my future endeavors. I would like to also thank Ilaria and Erica from the Education Outreach team for always being there when I needed assistance and for helping coordinate this program. I would like to further thank the various labs here at MPFI that helped facilitate my project with certain materials or reagents. I would also like to thank the various donors that allow programs like these to happen each and every year. And lastly, I would like to thank my family for being with me every step of the way. And now I'll open the floor to questions.
Questions for Jonathan?
Can you explain light sheet microscopy?
Yeah. So here. So the way light sheet microscopy works is basically a laser projects a beam of light into a few set of mirrors over mirrors and lenses over here that then reach a spherical lens that then condenses the light into a sheet of light. It takes the collimated light and turns it into a really thin sheet. That thin sheet is then passed through a number of other optics that control zoom and a few other things. And then that sheet of light reaches a pivot scanner, which is basically just a piezo crystal that flicks back and forth really quickly in like a span of microseconds to allow that sheet of light to then reach this prism on the left and right side. So then it reaches the 2 illumination optics and then feed into the chamber where your sample is. And then once it reaches your sample, the light is the excited fluorophores are read from your sample by the detection objective and then fed into the computer for live imaging.
Another question.
Hi, so you counted cells and we saw from the other presentations that knowing where the cells were was important. Do you have a way to analyze that, just like where these cells are in the brain?
Yes, so the question was, is there a way to analyze where the cells are in the brain as we've been segmenting them? So with the Arevis software, it actually offers a mapping a mapping capability where you can take a conventional map such as the Allen Atlas that's available, it's an open source available map, and add that to your data. And once you do that, it can see the location of each of your segments, if it's segmented cells or probe tracks or neurons, and literally tell you, as an example here on the bottom right, an example of how many segments are in each section of the brain. So as an example here, you can see how many cells are inside the insular cortex.